We've got two fantastic speakers today speaking on the topic of speech conditions and neurodiversity. So I'm hoping you can see my screen here. We've got Dr. Elsa Eising and Dr. Amanda Ludlow. And I've just got a few housekeeping things to go through first before I let Roderik introduce our speakers for us. So this webinar is hosted by the Intellectual Disabilities and Autism Working Group within AIMS2 Trials. And I just wanted to do a quick shameless plug of our group and some of the resources that we've created as a group. And if you want to check them out, if you haven't already, then you can access them via the link in this page. And you can also find them on the AIMS2 Trials website. If you want to find them. Everything we've created is available on the Open Science Framework as well for people to use and adapt as they need to. And then in terms of housekeeping, we have a few meeting guidelines that will help you navigate this webinar today. So managing our own noise during the webinar, you won't be able to unmute yourself or on your microphone or turn on your camera. That's for the first portion where we have our presentations. After that, in the second portion where we'll have a discussion, you will be able to unmute yourself and you will be able to share your camera, just to let you know. So please share your thoughts throughout. If you would like to contribute, particularly in this first portion, if you have questions or comments for our speakers, please put them in the chat. I will keep note of that. And then at the end of the talks, I can present your questions to the speakers. If you would like to personally present your question to our speakers, then I would ask that you hold on for the second portion of the webinar, where we'll have a bit of time to discuss before we go into breakout rooms, and you can pose your questions personally to our speakers then. The third thing is to stay engaged. So like I say, you won't be able to turn on your camera, but in the second portion where we have a discussion in little breakout groups, you can have your camera on or off as you prefer, but please get involved, speak in the chat if you prefer, or unmute yourself. It's absolutely as you feel comfortable. And then lastly, just to remind everybody that we obviously want to show respect and think respectfully. We challenge each other all the time on the things that we say, but we don't challenge people. Everybody has the right to feel that everyone has the right to feel the way that they do, and we respect that, and we can discuss difference respectfully in these spaces. And then lastly, being mindful of time. So we may have to move things on or move discussions on, particularly in the second portion, just to adhere to schedule to make sure that we finish at 2:30 as planned. So then a little layout of the webinar today. These times are a little bit subject to change, so please don't take them literally. But we'll first have our presentation by Dr. Elsa, and then we'll have a few questions and then a little comfort break. Then we'll move on to Dr. Amanda's presentation. Again, we'll have some questions and then a short comfort break. And then we'll come back after that and we'll have a little discussion and go into our breakout rooms. And then we can discuss some things. We've got some prompts for you to use in your rooms to help discussion flow. And then we'll come back together at the end for the final sort of 15 minutes. Just summarise what we've talked about in our breakout groups and try and go away with a few good ideas from the webinar. On that note, I will hand over to my co-chair, Roderik, who is going to introduce our first speaker.
Yes, thank you very much, everyone, and welcome again to this webinar about speech conditions and neurodiversity, particularly on stuttering and selective mutism. We're going to have two fantastic speakers today, and the first one is going to be Dr. Elsa Eising, Eising in Dutch, from the Max Planck Institute of Psycholinguistics in Nijmegen. Right after that, when she finishes, we will have Dr. Amanda Ludlow from the University of Hertfordshire. Dr. Elsa Eising is a geneticist, a specialist in the field, and Dr. Amanda Ludlow is a psychologist and also a specialist in her field. So please welcome Dr. Elsa Eising for her presentation. Thank you.
Yeah, thank you so much, Roderik, for this introduction. I'm going to share my screen. Let's see. I hope you now see my first slide. Thanks so much for the invitation to tell you something today about monogenic contributions to speech conditions. I'm very much looking forward to today and the discussion that's going to follow. I was asked to first talk about how I think about neurodiversity in my research. So as a geneticist, I look at the DNA, and this is sort of built during evolution. And evolution requires variation. This allows our population to adapt if the environment changes. And therefore, to me, it's normal that we're not all the same. The basis of these differences is genetic variation, so points in the DNA where we're not the same. And this causes us to be all different. But for me, it also provides an entry point for my research, where I'm trying to understand the genetic and the biological basis of speech and language. So in this talk, I will focus on rare variants with large effects, and those I can find by looking at people with speech conditions. But in the end, I hope that such variants help us understand these speech conditions, but also more general processes required for speech. So in here, I drew sort of a normal distribution that describes almost all the variation that we can find in almost all human features, where there is the bulk of people that look quite similar, but in the end, we can find people that are, well, slightly different from the average, if I can say it this way. Here, in my talk, I'm going to focus on people that are, that have certain speech features that you don't see in most people. In other work that I'm not going to discuss, we also make use of the full spectrum of variation that we can find in humans. So here I'm talking about sort of monogenic contributions to more extreme phenotypes. And I'm going to talk about three speech conditions today. The first one is going to be childhood apraxia of speech. This is a very rare motor speech disorder that is caused by difficulties in controlling the muscles involved in articulation. When you ask someone with childhood apraxia of speech to repeat a word, every time this person will make different speech errors. This is the characteristics of this speech condition. Next, I will talk about speech delay. Usually, speech delay is described as a later onset of speech. In the study that I did, we described speech delay as children who had significant number of speech sounds deletions and substitutions or errors that are abnormal for their age. Usually, they normalize with treatment. And then lastly, I will talk about stuttering, a speech condition characterized by specific speech disruptions, namely repetitions of sounds or syllables, prolongations of sounds, and silent blocks. So when you ask a geneticist what is the cause of speech conditions, or actually any human trait, they will say part of this is caused by the DNA, in addition to environmental factors from your sort of home environment or the bigger environment and random chance factors. So that's not a helpful answer because it applies to every condition. But in the case of speech conditions, I hope that the result of genetic research will help us a little bit understand speech better. As you're not all geneticists, I'm going to start with the basis, which is our DNA. Our DNA is organized in 46 chromosomes, and together these contain 3 billion base pairs or letters that contain the code to make our proteins and also describe when to make these. So, and as I already mentioned, not all of these base pairs is exactly the same in every person. Some will vary. And these genetic variants can be very big to very small, so they range from a single letter to a whole chromosome. They can be very rare, even unique to a single person, or they can be very common where about half of the people carry this variant. And everyone has so roughly 4 million DNA variants. So these are really many. These include so 45 to 90 DNA variants that are new, that you didn't inherit from your parents. So in the process of making sort of the egg cell and the sperm cell, new variations arise that everyone has, and sometimes they affect us more or less. This is a part of the evolutionary process that allows our full population to vary and to change. So my question here that I'm trying to answer is, which of these millions of DNA variants affect our speech, and how does they do this? So we can categorize DNA variants in a graph where we, let's see if I can have a pointer thingy that you can see, that we can split up on a scale from super rare to very common. And we can then on the y-axis split them up into based on the effect of the variant on us. And some variants have no effect at all, and other variants have very large effects. So you see that like all of these sort of this orange area is where variants can be found. There is a chunk missing, which is the very common variants with very large effect sizes. In general, they don't exist because of evolutionary pressures. So either variants are very helpful for us and then they will spread to the full population, so they won't be a variant anymore, or they're unhelpful and then they're pushed out and become very rare. So in genetic research, we can distinguish two types. And the first one looks at monogenic inheritance. And there we look for variants with a very large effect size. And these therefore tend to be very rare or even unique to a person. We can also look at complex inheritance, which I won't talk about in this talk, but in this type of genetic research, we investigate the common variants that have very small effects, and they add up together to affect our certain human traits. So when we look at, when we do genetic research, we usually start out with twin studies to estimate whether the DNA plays a role. In twin studies, we can compare monozygotic twins, so identical twins, and dizygotic twins, and we look whether the monozygotic twins are more similar based for a certain trait than the dizygotic twins. And if this is the case, then the DNA probably plays a role. For monogenic inheritance, we usually look in small or very large families to find a single cause for a disorder in the family or in an individual. And for common variants, we usually study those in very large cohorts, so I won't go into this any further. I do want to explain the rare and the rare monogenic inheritance and the complex inheritance once more, so you understand this difference perhaps a little bit better. So for complex inheritance, for example, when we look at speech can be normal, people can have a speech condition. This balance is determined by many DNA variants. Think about thousands of DNA variants in addition to environmental factors, and this will influence a balance whether speech is normal or whether there is a speech condition. When we talk about monogenic inheritance, there is a single DNA variant that explains the speech condition in a single person. And when we then look in the family, and here I drew the pedigree of a family with the grandparents, and then, well, the parents and the uncles and the aunts, and here the children and the nieces and nephews. And everyone marked in black is the family member with the speech condition. And you can see that the speech condition is quite common in this family. And in these type of families, we might find a variant in a certain gene that explains the speech condition in this family. So a single variant that is inherited here from the grandparent to the parents to the children that explains the speech condition in this family. Another family might have the same speech condition, but might have a very different variant in a different gene that causes the speech condition in this family. So even though this inheritance is monogenic, so in a single person, a single gene is involved and a single DNA variant, this gene and this variant may differ between families. And this is indeed what we see when in neurodevelopmental disorders. And of course, this is a very broad category. But so far, 1,500 genes have been identified that can cause neurodevelopmental disorders out of about the 20,000 genes that we have. So this is quite a huge chunk of genes, and these are all genes that are important, that play important roles during brain development. We also know that about 20% of individuals with the neurodevelopmental disorders have been described to have delayed speech and language development. But sadly, usually in the medical genetics, speech and language issues don't get better described than this. There is or there isn't delayed speech and language development. So we don't know whether these children, for example, don't understand language and have more of a language problem, or they might not physically be able to speak because of motor steering issues, or you can think of many other reasons for a speech or language delay. And these might usually actually be a rare disorder such as childhood apraxia of speech, or indeed a delay of language development that co-occurs with severe intellectual disability. What we also know of speech issues and neurodevelopmental disorders, that when there is a delay in the start of babbling and the first words, that there's an increased chance of having a genetic disorder diagnosis for a developmental disorder. So there is a very interesting link between neurodevelopmental disorders and speech and language. So for speech disorders and conditions, we don't have 1,500 genes that we have already linked to this. So the first link and the first gene linked to childhood apraxia of speech was found in this quite large family that lives in the UK, where about half of the members have childhood apraxia of speech. And they also have a DNA variant in a gene called FOXP2. This was the first time that a gene was linked to speech and language disorder. But we also know that this gene FOXP2 can only explain the childhood apraxia of speech in about 1 in 50 individuals. Well, there was one study done in 49 individuals, and they found only one FOXP2 variant in one of these individuals. So it's not a very common cause of childhood apraxia of speech. In 2010, the first gene was linked to stuttering in this very large Pakistani family. What the researchers, however, saw that this gene is more of a risk factor for stuttering. So the variant is more common in the individuals that stutter, but not always present. So the aim of my research is to identify additional causes for speech conditions such as childhood apraxia of speech, speech delay and stuttering, and also to use these genetic variants and these genes to better understand the biological processes involved and the genetic overlap with other traits and disorders. So in here, I mostly look at these de novo variants. So these are the variants you don't inherit from your parents. They don't have them, but they were newly arisen during the process of creating the egg cell and the sperm cell. Usually we have, as I said before, about 50 to 90 of these in our DNA, but about 0 to 4 per person in the part of the DNA that codes for the proteins. So this is a very powerful way to identify possible causal variants for traits that are present in the child but not not in the parents. And this is often used to identify causes for rare and severe developmental disorders. So what we did is to use this method and apply it on childhood apraxia of speech. And also what we did, you still get quite some variance. So we further filter to make sure that we only look at variants that affect proteins in a gene that don't contain DNA variants very often, so that are protected during evolutionary processes because they are important for us. And I only included variants that are in a protein with a role in the brain. And then I applied clinical method to classify variants as likely pathogenic. So this is very helpful to show that this variant is likely causal. It's a method that comes from the clinical genetics research. The issue is that it can only be used on genes that have already been proven to be a cause for a similar disorder. So you can't identify new genes for a disorder in this way. To identify a new genetic cause for a trait, you have to identify multiple individuals that have a variant in the same gene and a similar trait. My studies have usually a limited number of individuals, and therefore I never find variants in the same gene in different individuals. So I'm relying on this method to classify variants as pathogenic, and I will come back to this. That is because it's causing a little bit of a circular reasoning. This is because very few people have looked at the genetics of speech conditions. So I'm relying on genes that have been linked to neurodevelopmental disorders in the past. And therefore, the overlap that I see between speech conditions and neurodevelopmental disorders is slightly caused by using this method. Okay, now on to the research. I started out with a childhood apraxia of speech study where we included 19 individuals with childhood apraxia of speech, and 9 of them we also had DNA of the parents, so we couldn't look for de novo variants, and for another 10 of them we had to do some more strict filtering in different ways. So I'm for 3 out of 9 we could find a likely pathogenic de novo variant. For 5 out of 7, I found another 5 likely pathogenic loss of function variant that cause no functional protein to produce at all. And then there were 21 variants that are called missense that change only a small bit of a protein, and therefore the impact of these is very difficult to interpret. So I couldn't classify them as likely pathogenic. But still, in only 19 cases, I could identify 8 likely pathogenic or pathogenic variants. So 8 new candidate genes for CAS, and these were the first to be identified after the identification of FOXP2 gene in 2001. So this was a huge increase in the genetic knowledge that we had about this speech condition. So I used these genes to search for a shared biological function among the genes that we found. And I found that these genes are usually highly expressed in the brain before birth, and then sort of halfway through during gestation, and after birth their expression drops. So these are genes that are important for very early developmental processes. When we look at their biological functions, we see that they're mostly involved in binding the DNA and regulating the expression of other genes. So these are genes that play a role in sort of orchestrating the development of the brain. So later on, and already before my study, these genes have been linked to other neurodevelopmental traits. The CHD3 gene was linked to a neurodevelopmental trait actually as a response to my research, and the others were already linked before to neurodevelopmental traits. And I'm just showing you the headers of papers that have linked these genes to neurodevelopmental traits, and you can just take for granted that they did. So this is a little graph that shows the progress of our genetic knowledge on childhood apraxia of speech during time that started out in 2001 with the identification of FOXP2 and then a whole lot of nothing during the 18 years to follow. And then my study was the first to identify another 8 genes that can cause childhood apraxia of speech. So later on, other studies have identified another bunch of genes, and now we have over 30 genes that we know of that can cause childhood apraxia of speech. And most of these play a role in very early brain developmental processes. What we also know is that these genes, so here you find a list of all of these genes, can this cause childhood apraxia of speech, but they can also cause intellectual disability or autism spectrum disorder, quite many of them. So the majority can also cause epilepsy. They've been described to cause ADHD, cerebral palsy, and schizophrenia. So these genes don't always cause childhood apraxia of speech. In different individuals, they might variants in these genes can cause very different features. So that's a bit the current status of what we know about childhood apraxia of speech. And I'm now switching to speech delay. So in this study, I looked at 23 children with speech delay. For again 10, we could include the DNA of unaffected parents to look for de novo variants. So these children were aged 3 to 9, and we selected them to have no intellectual disability, because previously in the work on childhood apraxia of speech, sometimes the children had a more complex features than only a speech condition, and they might also have a lower IQ, for example. And then, well, the question is, are you really studying the speech trait, or are you actually studying a more broad neurodevelopmental disorder and just in a very small study? So here we try to avoid this by including only children with an IQ higher than, well, 83 was our lowest number. And again, we looked at de novo variants and variants that fully disrupt the protein and variants that change only a very small part of a protein. And we could identify one loss of function variant that I could classify as pathogenic and 3 missense variants that are likely pathogenic. So in this case, we couldn't find a de novo variant that seemed to be causal, but we identified the first 4 DNA variants and genes that can cause childhood speech delay. One of these is SETD1A, and that's already linked to schizophrenia and a more broad neurodevelopmental disorder. And actually, this gene was also previously found in my study on speech in childhood apraxia of speech, and another study on childhood apraxia of speech as well. So this gene seems to be like quite speechy, if I can say it this way. What we also saw was that the child that carried the frameshift variant, the loss of function variant in this gene, was the most severely affected of all the children in this cohort. Yeah, that was this one. And then quickly on to stuttering. Here we had data of 84 trios, so children who stutter and their parents who don't stutter. And stuttering is something that sometimes persists into adulthood, but often resolves during childhood. We included 47 children with persistent stuttering and 37 with resolved stuttering, or where we weren't completely sure whether it was fully resolved. We made sure that the parents didn't stutter and that there was also limited stuttering in other family members. And here we could identify 4 variants that we could identify with a likely or a pathogenic variant, and 2 genes of interest. So these haven't been linked to neurodevelopmental disorders yet, but have a variant that looked very interesting and impactful. And so we had 4 or 6 new candidate genes for stuttering. Because we had parent data for all of the children, I don't have separate categories for the loss of function variants and the missense. And again, we found genes that are also causal for neurodevelopmental disorders with broad features. Here I'm again showing the parts of the articles. So these cause, again, neurodevelopmental delay, cognitive impairment, etc., etc. With the genes previously found for stuttering and the 6 genes that are now implicated in stuttering, I tried again to search for an overlap in biological function among the genes to learn more about the processes that cause stuttering. I tried different approaches, including the one that I used for childhood apraxia of speech that was very successful, but I couldn't find any overlap in biological function. So in that sense, stuttering is very different from other speech conditions. Although, at least for now, for the first time, I was able to link it to genes that also cause neurodevelopmental disorders. Here I want to talk a little bit about the diagnostic yield. So when children have a speech condition and you do genetic research, for how many of them can you find a cause? And for childhood apraxia of speech, in the different studies, including mine in 2019 and others done later on, on average, we can find a genetic cause for about 30% of the children. This was quite different for speech delay, where we found a genetic cause for 13%, and for stuttering, especially where we found a genetic cause for 5%. This is also perhaps what we expect based on the rarity of the trait. So childhood apraxia of speech is a very rare condition and it can be very severe, while stuttering is very common and is probably genetically much more complex and less often monogenic. And a speech delay might a little bit be in between these two. When we look at the genes that we have identified, we are starting to see a little bit of overlap among the genes that we implicated in childhood apraxia of speech. This is where we know most of the genes, where most of the genes have been identified. We can see 3 genes now that overlap between the 3 studies. And one gene overlaps between speech delay and childhood apraxia of speech. So when we add stuttering to this, we see one gene that overlaps with speech delay, which is quite surprising given that there's only 3 genes identified so far. And I actually know that in a study that a collaborator is carrying out, that this gene here is also just identified as a cause for childhood apraxia of speech. So it's now implicated in all 3 of these speech conditions. So what I think is happening is that when children have a neurodevelopmental disorder and are very severely affected, they will go through clinical genetics testing. And then they might also contribute to studies on neurodevelopmental disorders that have been published in the past. When children have only speech features, they will go to a speech therapist. And because speech therapy is organized separate from medical hospitals, they won't be approached for genetic testing.
Sorry to interrupt.
This is the type of children that we see in our studies on speech conditions. And what we're finding now is that they actually are caused by the same genes as the severely affected children with neurodevelopmental disorders. So we're probably capturing the much more mildly affected children. So how a single gene can have different outcomes in different individuals can be caused by the specific variant in the gene, but also another the genetic background or in the environment. So the same variant can have a very different outcome on different people. So what we're doing here is that we are expanding the knowledge about what, so the clinical picture is that you can expect from a mutation in a certain gene. So what colleagues in Australia have also been doing quite actively is now go back to the patients with neurodevelopmental disorders that are caused by the genes that we see in our studies, and then characterize the speech phenotypes in them. And she sees indeed that many of the children that have these neurodevelopmental disorders also have speech conditions or motor impairments, but that sometimes underlie the articulation difficulties in childhood apraxia of speech, for example. So what we are doing is that we're probably identifying the genes that cause neurodevelopmental disorders, where speech is a quite major feature of the disorder. And actually, in these children that have a more broad neurodevelopmental disorder, the speech difficulties are often pointed by parents as the most, as those that affect the children most and also the parents most, because they make it very difficult to interact with each other and to understand each other. And therefore, the speech features, out of all the features associated with these neurodevelopmental disorders, might have most impact. So in conclusions, I hope that I showed you that speech conditions sometimes have a monogenic cause, and we're starting to find genes that more often cause speech conditions, where probably many genes left to find that can cause speech conditions, because otherwise we would have found the same ones over and over again. We're finding genetic overlap between speech conditions and neurodevelopmental disorders. And because of this, we're broadening the phenotypes associated with these neurodevelopmental disorder genes. And for some neurodevelopmental disorders, speech difficulties might actually be a central component. And I believe that we're finding those neurodevelopmental disorders and those genes. And we might miss genetic causes for speech conditions that only cause speech conditions and not neurodevelopmental disorders. And that's because we only can classify variants as pathogenic or likely pathogenic when they have already been linked to a disorder. So it will take a while before we have investigated enough people with speech conditions until we find genes that we see more often only in the speech conditions to build proof that these genes are indeed involved. So, and this work was done with many other people involved in subset of the different studies that I explained to you, and especially my colleagues at the Max Planck Institute here in Nijmegen. So.
Now we will jump to Dr. Amanda Ludlow, who is from the University of Hertfordshire, as Roderik has said, who's going to talk to us about selective mutism.
Hi. So I head up the what we call the Psychology and Neurodiversity Applied Research Unit. My background is interested in autism and all different types of neurodiversity, from tic disorders and pathological demand avoidance. And I'm currently the National Institute of Health Applied Research Collaboration East of England theme lead for the mental health and neurodiversity. Just very quickly, I'm coming today from a kind of trying to tell people's stories. It comes from approach working with professionals, educationals, charities, people living with different symptoms, and trying to tell their stories to advocate and to get them the best support. And with that, we work with amazing bunch of collaborators within Panda. The symbol of Panda represents this idea of an idea that when people say panda about the black and white panda and the fact that pandas can be read, that we are all similar and we're all different. And it's about celebrating those differences and actually how we access support and advocate for that. Regarding selective mutism, what I'm going to try and do is a whistle-blown tour of some of the studies that we've been looking at it and trying to highlight what is selective mutism and what we know about selective mutism people that show autism as well. And the fact, much like with the previous speaker, there is this idea that actually speech conditions are very under-recognised in children with autism, and there's very limited treatment and support and even research really in comparison to other areas. And hopefully we'll highlight that today and where some of the parents have raised where they see the priorities for research. Selective mutism, in essence, is an inability to speak, respond in certain social environments or situations. So, for example, at school, or with someone that's unfamiliar, despite being able to have perfectly well-spoken language in a home environment. Parents often describe it as being like a communication freeze triggered by the environmental people, whereas the Royal College of Speech and Language Therapists have defined it as speech phobia, where someone gets a lot of anxiety and physiological reaction, almost like freezing, leaving them unable to actually speak. But currently, diagnostically, it's recognised as an anxiety disorder specifically related to social anxiety, because that is highly co-occurring with children that present with selective mutism. Although to get a diagnosis of selective mutism, you're supposed to show people show consistently fail to speak in specific social situations expected. You must have, or a lack of speech must interfere with the ability to sort of cope and be present for at least 1 month, and it should be differentiated from other communication difficulties. What we don't know is whether selective mutism disappears. There's very few longitudinal studies following up children into adulthood, and where they have been done with adults, it's suspected that actually the social anxiety aspect of selective mutism certainly remains. But actually, despite the fact that a lot of the onset of selective mutism is very early, most children won't get picked up with selective mutism until they get to primary school. Just giving an example of some of the types of questions to get you thinking about what selective mutism might look like. So often that people use a selective mutism questionnaire as part of the diagnostics to pick up, and it's looking at how people respond in different situations. Quite often children will go and be seen as very shy, they don't like public speaking, they have very kind of tight group network of friends. Parents find that they are much more comfortable with familiar adults. But one of the difficulties that we have with things like selective mutism, we know like between 20 to 50% of children with selective mutism will show language difficulties as well. So despite it being one of the criteria for being, it must be, has to be differentiated, we do know that they have problems with expressive and receptive speech. But most of the time you'll find a very high predominance of selective mutism, specifically in the school environment, which is quite often that teachers are sometimes the first to kind of pick up on some of the symptoms of selective mutism. And it's thought to be as high as 1% of the population, but we know that there's so many overlapping conditions with selective mutism, and because of people kind of linking it to anxiety disorders and or shyness, it's quite often difficult to actually get people to present. And as previously discussed, there's very few speech and language therapists that are kind of working alongside sort of neurodevelopmental services to actually pick up when they are co-occurring conditions. Know that a very high proportion of people with selective mutism will reach the criteria of social anxiety disorder, and actually a lot of studies that have compared the two groups together show that they have as high levels of social anxiety as those presenting with social anxiety disorder specifically. But they also have auditory processing difficulties, and there's one suggestion that actually part of the problem, one of the difficulties children with selective mutism has is actually they don't like the sound of their own voices because of the auditory. So that self-vocalization triggers this negative impact on leading to restriction or avoidance of speech patterns. There are two very different types of what we call profiles that sometimes are linked to selective mutism. So the high profile selective mutism is characterized by young people who are unable to speak to all certain people, making them most observable because they're very restrictive in the people that they speak to, making sort of just everyday type tasks at school very challenging. But there's also the low profile selective mutism, where they'll occasionally speak when prompted, often regarded as shy, quiet, but people don't recognise that you can actually swap between the two presentations, and both of them are seen to be as highly intense in terms of the anxiety it provokes. And this lack of understanding often means that people who are considered in the low profile selective mutism are less likely to be picked up or given support. There are lots of questions about, is selective mutism anxiety as it's currently reckoned in the diagnosis? Is it neurodiversity? Because so many children who present with selective mutism will reach a diagnosis of autism and or ADHD. There's some research to suggest selective mutism may conceal some symptoms of autism, but studies of up to 80% of people with selective mutism have reached diagnosis of autism, showing that there's such a high co-occurrence between them. There's currently no clinical guidance, and the previous diagnostic for which was used to diagnose DSM, DSM-4, actually autism was an exclusion criteria, so you couldn't get selective mutism and autism together. Now you are, but there is a real knock-on effect of people who are still unable to access one or more of the diagnoses because they either don't reach the threshold for either, or because they're presenting with both symptoms, one disorder is given over the other. And there's such little research about the co-occurrence of these two conditions together. We also know that females, and there is a higher prevalence of selective mutism in females, but because with a lot of the research showing about masking and coping in social situations, they particularly might get missed in terms of selective mutism as they try and compensate, but actually are struggling. And actually, we have found in some of the research that we've done that there is a difference between selective mutism and autism diagnosis in terms of the ages at which they present only in the females, that they have a massive delay in the autism diagnosis once selective mutism has been picked up, where we don't see the same with males. So there are potentially vulnerable groups within this selective mutism that we're still really unsure about what and how they compare. We did some studies trying to look at what selective mutism is and what selective mutism with a diagnosis of autism might mean. And this is just a small set of participants. We've actually tripled the number of participants that we've got, but the same patterns remain. And we had fairly balanced samples of people with selective mutism and selective mutism of autism. Questionnaires were looking at selective mutism autism, and to get into selective mutism autism, you had to reach the criteria. Most of the participants, all the participants with selective mutism were diagnosed, and the ones with selective mutism and autism, some of them were awaiting diagnosis because they were already diagnosed with one, not the other. And we're looking at it in relation to anxiety and depression. And some of the problems that we found in terms of actually how you think about selective mutism and autism is the fact that there are very high co-occurrence between what is selective mutism by itself versus those with selective mutism and autism. The only factors that we kind of found that differentiated children from the selective mutism with those with the co-occurring autism was whispering. So whispering was very specific to selective mutism group only. Whereas with the children with the autism, we found a little bit more about avoiding social situations and complaining of things like stomachache with the anxiety. But actually, some of the key symptoms of selective mutism are so overlap between the two, it shows why it's quite often difficult to distinguish between them. We also found that there was no difference in the age onset between selective mutism with those with and without autism. So they were showing from the parents' perspective early signs, both regardless, and those selective mutism signs became before often autism symptoms were to be shown. But children with selective mutism, unfortunately, were shown to have quite positive relations with peers and teachers and professionals, where we weren't finding the children with autism to reporting to the same degree. So we found some of the characteristics that distinguished this group with the autism specifically was they were more likely to have language delay. There was a massive gap between the onset of the selective mutism behaviours and an actual diagnosis of selective mutism in the autism group compared to selective mutism by itself. They were more likely to report more negative experiences with peers. And importantly, even looking at the selective mutism questionnaire, those with selective mutism and autism showed high severity selective mutism symptoms, high levels of stress, showing that there's really importance in understanding how selective mutism within this group. Looking at co-occurring, again, we didn't find a huge amount of differences, but you were more likely to have additional mental health concerns in the selective mutism group only. Family incidents also didn't differentiate the two groups dramatically, other than selective mutism autism. There was a family of autism, history of autism in the family, and also learning disability in the selective mutism and autism group. But other than that, there weren't many differences in the way the families reported sort of a history. And the biggest difference in terms of treatment was the fact that selective mutism without the autism, you were more likely to access psychotherapy, and you were actually more likely to access treatment in general compared to the group that actually had selective mutism and autism, showing there's a group that are just not accessing treatment. One of the key things that we were suggested that might differentiate the autism and selective mutism group was sensory difficulties and how they kind of take in the information. So we were interested to see how that related to anxiety, but between the two groups, because there's quite complex interplay between what is anxiety and sensitivity. So when you have lots of information in your environment and it can be very anxiety provoking, we certainly know in children with autism that can elevated levels of anxiety affects kind of the want to do social interactions further, and may be how they manage that distress can be through withdrawal muteness, as in we've seen with selective mutism, or disengagement with social situations that sometimes children with autism show. And we just did a small study where we're looking at 75 parents. Half of them had autism and selective mutism, the other half had pure selective mutism, again using similar questionnaires and a sensory profile. One of the things we found that was the children with autism and selective mutism were more likely to have a diagnosis of general anxiety disorder and social anxiety disorder, which we didn't find in the selective mutism group by itself. But the key thing is what differentiated the two groups was sensory avoidance. So children with autism and selective mutism are more likely to show sensory avoidance, and that relationship between social anxiety is predictive of that group only. So that it's suggesting that the sensory difficulties that children with autism sometimes have may differentiate it from the group with children with selective mutism per se. But the importance is the impact. One of the stories that's just not coming through is the impact of living with selective mutism and autism, particularly when you're struggling to get a diagnosis for either of the conditions. So we were just doing some qualitative interviews with the family and asking them about their impact, their experience of communication with health professionals, including schools, what the assessment process has been like, but also advice that they would have to other parents. And some of the key things that came were about the complexities, that interaction between the sensory difficulties that the children have when presenting with selective mutism and autism, and how that manages everyday life, but also the fatigue and communication freezes and shutdown that the children have in everyday socials, but also the impact it has on the family, affecting every aspect of the children's life, the siblings, but also parents' ability to cope with it. Just want to kind of focus briefly before we finish on 3 quotes that came out that were quite useful. These are all from parents with children with selective mutism and autism. So one was suggesting it took over 10 years to get an assessment because they couldn't find a speech and language therapist who is specialism in selective mutism, despite the fact that they were diagnosed at 3. So by the time they got a speech and language diagnosis, a selective mutism diagnosis, they were 13 years of age or in secondary school. And another parent talking about the sort of sensory demands on child with selective mutism and autism, who described this grandma who's had pain in her arm, and the child asked where, but instead of the grandma touching her own arm, she touched the daughter's arm, and the daughter instantly shut down. She stopped speaking because she'd been touched and she wasn't expecting it, and she just couldn't cope, highlighting just how extreme this anxiety is and the importance of understanding it, but also then what support can we provide for children to be able to cope, and help support parents in advocating their process, the pathways forward. And I think the one thing that came through from the parents was just the direct impact it had on everything. So there was also about the siblings, the fact they couldn't have children coming around to the house because the child with selective mutism really struggled to have people in the house, and how debilitating that was in terms of the social opportunities that they could have for their younger or older siblings. So the main conclusions, really, what we're currently looking at is there does seems to be such a limited understanding about speech and language generally, I think, in autism. But I think in relation to things like selective mutism, there needs to be clearer understanding about the way it progresses through development, more longitudinal studies, its relationship to social anxiety, who's responsible for diagnosis, and how that distinguishes between different presentations, such as males and females. But there is such a significant delay in diagnosing selective mutism when autistic traits are present, and that is really important, therefore, that we find better ways to support people that are navigating that diagnosis. And to finish with my beautiful red panda.